Hi, biology students. I'm going to continue on talking about some parts of the cell and then move into cell theory a little bit. So this is where we left off. We said that plant cells are different than animal cells in these three ways, which means that just for clarification, all the other things we talked about as we went through parts of the eukaryotic cell, just for a quick review, um, plant cells and animal cells have cell membranes. They all have mostly the same organelles like mitochondria and ribosomes, endoplasmic reticulum, Golgi apparatus, these vesicles and lysosomes, cytoskeleton. Plant cells do not have cilia and flagella. That is one thing that is only for mostly single-celled animal cells, and we also said that there's cilia on the cells that line our respiratory tract. Usually cilia and flagella are used for movement of cells, and we talked about how the cilia on the cells in our respiratory tract are more for trapping those particles that shouldn't be in there so that we sneeze or cough them out. So these we don't find in plant cells, but the rest of them we do. Nucleus are in both and so forth. And then plant cells have some other ones that, and again, we already mentioned this. So plant cells have cell walls, vacuoles, and some plastids like chloroplasts, but there's other types of plastids as well. So plant cells have those cell walls. And I think we did talk about this too, kind of the different structure and so forth. Vacuoles in plants, we said animal cells can have vacuoles too, but in plants they're different because they're very, very large. They can take up to 90% of the cell might be composed of this large vacuole. And we see when this is really, full of water wouldn't be pink, that'd be weird. If this is really, let's draw our water blue. If this is really full of water, then it actually makes the plant cell kind of plump, if you will. We call that turgid. So turgid means that those vacuoles are full of water and they're pushing out on the cell membrane. Think of it like an inflated balloon. So if a balloon is really full, then it's more turgid. If a balloon is completely out of air, then it's it's just that empty balloon. There's no air pressure in there. So we actually have water pressure pushing against the inside of this cell. It actually pushes the cell membrane toward the cell wall. So again, if it's full of water, it also contains some enzymes and some waste as well. But if this vacuole is full, we say that the cell is turgid. What do you think happens if these vacuoles are not full? So what happens to plants when they don't get enough water to keep their vacuoles full? So if it's not full, we would notice the plant would be wilting. Eventually the plant would die if it didn't get enough water, but it can exist in that wilted state for quite some time. Other types of plastids, we mentioned plastids are one of the organelles that plants have that animal cells do not have. Leucoplasts is one. Leucoplasts store starches, lipids, or proteins, and these are mostly found in the roots of plants. No photosynthesis is occurring here, so they wouldn't be green like other parts of the plant. So if you think of potatoes, potatoes grow below ground, as do carrots and radishes and those kinds of things. But think of carrots. Carrots are very high in starches, and they can store these extra starches. They don't need to make room for the chloroplasts where photosynthesis occurs, but it is important that the plant be able to store nutrients to be used later when maybe there's not enough sun to be really active with photosynthesis. And then chloroplasts are the plastid that we think of most often. This is all about photosynthesis, and this is, of course, unique to plants. Animals definitely, I don't care how long you stand in the sun, you are never going to be photosynthetic. So this is all about plants, and this is how ultimately our world, harvest energy from the sun. It all is because of plants, because they have these chloroplasts with this magical substance called chlorophyll inside of it that captures energy from the sunlight. And we're going to talk about this process in a lot more detail later in a subsequent unit. So I'm not going to spend time talking about that process now, but they are just these incredibly magical organelles that can harvest energy from the sun. And again, they are the only things that that can do that. I'm going to go back and talk a little bit about, because we skipped ahead to organelles, I do want to go back and talk about discovery of the cell, because this is really important. So this, I know this slide is a little hard to read, but these are obviously plant cells. We know because they're box-like in structure, and they have all these little green dots, and I think by now you know what these little green dots are. So Robert Hooke was the first person way back in 1665. He examined a thin slice of cork. So cork actually comes from, hey, I'll show you a little video on that after a bit. Cork comes from the cork tree. It actually is a special kind of tree that makes cork. The harvesting process is really cool. But anyway, back in his day, they didn't have a lot of other ways to close jars and bottles. So kind of everything was closed with different sizes of cork. When he looked at it, he noticed a whole bunch of these little little box-like structures. And he's like, well, that's interesting. It was a very unique structure. I also want you to think about how come nobody before this time, they had used cork for centuries before this, how come nobody else ever noticed that? What had to be invented before you could look at these tiny little structures? Please think about that for a moment. And then he noticed in these little tiny structures, and he said, hey, these remind me of the little rooms that monks at monasteries sleep in, and those were called cells, and so he named these cells. They were present, interestingly enough, in all the plants he looked at seemed to have these same cool little structures. Sometime later, a Dutch scientist by the name of Anton van Leeuwenhoek observed some animal cells. He actually took something and scraped this is really gross, but the scum off of his teeth. They didn't have great oral hygiene back then, so he took scrapings off of his teeth and looked at it under a microscope, and he observed that there were tiny little, he called them wee animalcules. They were actually bacteria, but we now know, and the connection wasn't made that, oh, these are kind of the same thing that we find in plants when we look at them. So he didn't recognize that they were the same type of cells that Robert Hooke saw. So he gave them a different name. He called them wee animalcules. It wasn't until sometime later that some other scientists took all of the known information about cells and came up with 3 parts to the cell theory. So they came up with, and again, this is a couple centuries later in the 1800s. So I'm going to say this is in the 1800s. Sorry, I'm writing with my mouse. It's super messy. So this is in the 1800s. And they said all living things appear to be composed of cells. And we talked earlier in the year here about, yeah, cells seem to be present in all living things, and that's one of the criteria of living things. And they're not present in non-living things. So all living things are composed of cells. Cells are the basic building blocks of living things. So the basic building blocks of living things. All of our tissues, for example, are made of cells. Our hair made of cells, our teeth made of cells, our bones made of cells, and so forth. So they're the basic building blocks of all living things, whether we're talking about plants or animals. Cells only come from the reproduction of existing cells. So we don't find, again, we can't, even though we know the molecular components of cells, we can't mix up a batch of cells and give it the right conditions, and actually have cells spontaneously form, that is just simply not possible. And your body contains over 200 different kinds of cells. Each cell is specialized, or we say differentiated, during stages of development. In the very beginning, a fertilized egg the cells begin to divide, and in those early stages, that mass of cells are all identical. There's no differentiation. But very early on, differentiation begins to form. Cells in our bodies are all different sizes and shapes. The longest cell we know of is in the leg of a giraffe. There's a nerve cell that's about 2 meters, which is like 6 feet long. But there's lots of variation in shape, and we're going to talk about a few of those different ones. Nerve cells do tend to be very long. If you think of the job of nerve cells, they're responsible for sending these electrochemical signals from, let's say, from your fingers. You touch something hot, and it has to tell your brain that registers that, oh my gosh, that's really hot, what should I do? And the brain has to tell the muscles to contract and pull away from that source of incredible pain. And so the faster they can send those messages to the brain and spinal cord is incredibly important. So nerve cells tend to be very long and thin. There's something called the cell body that kind of interprets, but then it sends these messages, and we call this the synapse, from one nerve cell to another, and it's this big long chain all the way to the brain and spinal cord. So their structure is very related to their function. The longer they are, the more efficient, the fewer times they have to send it to another cell, and the faster that they can transmit that message. Skin cells are very flat. If you think the job of skin is to provide a protective covering for our body, and that's important. So they can cover more surface area if they're flat rather than round. White blood cells can change shape. This allows them to move through very narrow openings to isolate and digest bacteria and other foreign particles, and that's a really important part. All cells can be categorized as either prokaryotic or eukaryotic. You are made of eukaryotic cells, and so are plants and algae and funguses or fungi. These all contain a nucleus. They have organelles that are surrounded by membranes, so we could say membrane-bound organelles. They're more complex than prokaryotic cells, and they're found in all multicellular organisms. They are much larger, more complex. They might be multicellular, but they also can be single-celled or unicellular. The prokaryotic cells, then, these do not have a membrane-bound nucleus. They're this, smaller cell shape. They have a nuclear region where the DNA kind of hangs out, but it's not like our cells, which are eukaryotic and much larger. We have this nucleus, usually near the center of the cell, and this contains our DNA wrapped in a nice protective membrane-bound organelle. So they do not have membrane-bound organelles. When we talk about prokaryotic, we are only talking about bacteria. So all bacteria are prokaryotic, and all prokaryotic cells are bacteria. They're classified in different kingdoms from the eukaryotic because they're very different. Although their genetic material, that DNA or RNA, or DNA, well, and RNA, I guess, just sort of hangs out in this nuclear region. So they're always single-celled. You are made of eukaryotic cells. That's a good way to remember it. And then just really quickly, this is the hierarchy of living things for multicellular organization. So cells are the simplest unit in all living things. Cells make up tissues. Tissues make up organs. Organs make up organ systems. And then organ systems make up organisms. So A group of cells working together to perform a common function makes up tissue like muscle tissue.